that route. And this is why for rabies it's very difficult to use a blood test as an indicator of the infection. So blood tests are useless in case of rabies because in this case, first of all, the virus is not present, and also the antibody to the virus, they seem to cross-react with other viruses. So they're not really specific to the rabies virus. Okay, so how do we know how viruses are spread, and what is their movement, and what's the time they take? It takes, if you want, between infection, the start of an infection, until the symptoms appear. One small experiment was done by Fenners and mousepox to get an idea about the pathogenesis of viruses. And basically, what he did is classified, or he introduced, if you want, the virus in the pad of the animal, and then follow this, the presence of the virus in, if you want, in the bloodstream as well as in various organs. As seen here, in the case of the mousepox, and you have to remember that varies a lot between viruses. So if you want, this pathogenesis can be different, first of all, among different viruses, but also how the species, if you want, or the animal react to the virus or resist the virus. So day 0 to day 2, it took about 2.5 days for the virus to appear in the blood. So that's from the foot pad, okay, injection through the foot pad, and it appears in the blood after 2.5 days. And then, about between 3 and 4 days, the virus appears in the spleen and liver, so uses the blood to spread to various organs. And then by day 6, some, if you want, indication of the pox, if you want, the pox symptoms of the signs appear on the skin of the animal. And it took not until 11 days after the virus was introduced that the mouse exhibited severe rash and ulceration. So that's typical, if you want, of the mouse pox, but that's a mouse pox which would be, the sheep pox is going to be different in pathogenesis. Capri pox is going to be different. So that depends on obviously the species, and depends on the way the animal resists the infection. Okay, so now how viruses introduce injury or cause injury in the host. There are different ways. For example, we talked about the cilia movement as a way of preventing, pathogens from settling down on the epithelium. So viruses can cause the abrupt cessation of ciliary function. They can do this through different mechanisms, but basically when this is done, then viruses can reach, obviously, the epithelium, settle a certain non-specific receptor in the epithelium, and enter the epithelium to the subepithelium layer of the respiratory tract in this case. Now, also they can stop, for example, the exudation of inflammatory fluid. They can block the airways by causing, killing some cell, causing cell debris, that block the airways. By affecting regeneration of the mucosa. For instance, an example in the gut, let's go to parvovirus. It kills Crick cells, and Crick cells in parvovirus, what's the function of those cells is generating the columnar epithelial cells. Without generating the columnar epithelial cells, obviously the epithelium is going to be dysfunctional. And most viruses act as a way to predispose the host to bacterial infection. So, an example, kind of cough, adenoviruses, distemper, and other viruses can predispose the animal to the bacterial infection by the bacteria Albophtella bronchoseptica. And the GI, again, it's the same. We talked about, killing cells and preventing the epithelium from forming. That obviously will create an imbalance in electrolyte, and hence the diarrhea, in the case of parvoviruses. And as a result of diarrhea, you have dehydration, acidosis, hypoglycemia, or they can cause a general malaise if they breach the intestine or the gastrointestinal tract. They go systemically to other organs, and hence they cause the malaise, if you want, in animals. And they predispose, like in the respiratory tract, to infection, bacterial infection, secondary bacterial infection. Now this is some of the mechanism of viral injury. Erythema, which is, which is reddening, if you want, of the skin. The papules, they're usually raised features on the skin. The macules, they are usually flat and reddish, if you want, in color. And the pustules, when the macules or the papules become infected, then there is pus, hence they are called the pustules. Now, there are ways that viruses escape, if you want, the host resistance. And they do this by different ways. Now, first of all, they can block synthesis of host DNA and RNA. Now, we know that for synthesis of host DNA and RNA, there are some enzymes that are needed for this. In addition to other things that participate, if you want, in forming a DNA or RNA. So viruses can block this mechanism. They can block host transcription. Parvoviruses and reoviruses are good in doing this. They can block further stages. Messenger RNA, for example, production of proteins. So they can affect the production of proteins, and they can interfere with the cellular membrane function by either punching holes in the cell membrane, lysing the cell membrane, and so forth. Now, we know that viruses can cause also neoplasms. And the association of viruses with cancer has been known, for a very long time. Now, for example, papillomas in dogs, herpes, are can cause some kind of, they can cause carcinomas. We know the sarcomas, which are, cancer, if you want, derived from the mesenchyme. Retroviruses, FELV, can cause sarcomas. And some viruses, like FALV, can cause also, and Marix can cause lymphomas. This is just a photograph of thymic lymphoma in the thoracic duct in the case of FELV. Now, how do they do this? And this is just a picture of papilloma virus, sarcoid in a horse, and the squamous cell carcinoma in the dogs and cats. So the question is how viruses do, yes?
I'm sorry, did you upload these? I don't have these slides, so
You should have it because I updated the...
you changed it.
Yeah, I updated it. So how do they cause, how viruses cause, if you want, cancer or neoplasm in general? Now we know that all animals, including us as well, we have what's called the proto-oncogene. And the proto-oncogene, they are genes that are normal. However, they can become abnormal and then cause cancer. And what makes them abnormal is the susceptibility to certain factors. That's in general. So we know, for example, radiation, chemicals, UV light, can make changes in the proto-oncogene and to make them oncogene, which are cancerous. So viruses, they exploit, if you want, this. There are two ways viruses can cause cancer. The first thing is what they can do, they can pick up the proto-oncogene from the cell that they infect, and later on they introduce the mutations in the proto-oncogene to make it an oncogene. So once they infect the cell again, they introduce the oncogene into the cell and the cell becomes cancerous. Or we know about retroviruses. Retroviruses, they integrate in DNA of the host. So they can carry, by integrating, if you want, next to a normal proto-oncogene, they can introduce changes to that proto-oncogene to make it an oncogene or cancerous. So there are various ways viruses can introduce those oncogene into the host. Did we understand this? Yes?
Yeah, would the P53 be an example of either the proto-oncogene or the oncogene itself?
The oncogene.
Oh, it's the oncogene?
Yeah.
Okay.
P53. And this is just a list of common mode of transmission of viruses. It's just for your information. At this stage, later on when we come to diseases, we're going to see all the mode of transmission as well as other things. Okay, so how do we control viral infections? We know that viruses do not respond to antibiotics, and we know that antiviral drugs are still not in so much use in for animals. So obviously the best control is to have an epidemiological study that can guide us towards management control. And to assess the, we need to assess the virulence of a particular pathogen or particular virus. So we need to know how many animals are affected, how many dead animals are affected, whether there is a certain factor, whether it is in the farm, in the environment of the animal that can cause this, whether the animals have been imported from abroad and we're introducing a new infection to the health, etc. Why do we need this? We need to know this so that we can introduce control management, if you want, projects or approaches, so that we can implement them and introduce surveillance mechanisms. Now, obviously, vaccination is important, and vaccination limits the spread of infection. Vaccination, when it comes to herd immunity, and what we mean by herd immunity is that, take the example of, for example, FELV and FIV, particularly FELV vaccination. Since the introduction of vaccination against FELV, the spread or the transmission of the infection has been reduced dramatically. There are still, and that's what we call, if you want, herd immunity. So basically, is to break the transmission cycle of an infection by introducing something like, vaccination. There's always the issue of whether there is contamination or there is unhygienic and whether there are, farms are infected, they spread infection, there is no hygienic rules being followed, and so on. So hygiene and sanitation are another way of, for breaking, if you want, the transmission cycle of viruses. The other way is what's called all-in, all-out management. And again, the all-in, all-out is an example of this is, for example, finishing, breeding and growth of animals, and then introduce hygiene and sanitation procedures into the farm before importing new animals or introducing new animals to the farm. Another way is separating the young from the old because of the susceptibility of the young to acquire infection from older animals. Another way is to separate, for example, animals that are pregnant from non-pregnant, and to facilitate delivery in private rooms rather than in a room where animals are mixed, and so on. So basically, separation, disinfection, and repopulation. Arthropod vector control is one way of limiting viral infection, although this is really not that practical. So other modes usually of limiting the spread of infections are followed. And obviously the surveillance, the quarantine, when it comes to surveillance, for example, we can follow, if you want, the presence of population of mosquito in certain regions in the world, or in in specific region let's say in the US. So there are surveillance programs, for example, for West Nile virus, where it can predict, if you want, the number or the density of mosquito in certain regions. So one can then adopt, if you want, some approaches that can limit infection of these animals, in putting them in different regions. Now, I want just quickly to introduce this term. Now, we have all heard of endemic and epidemic. In animals, we use enzootic and epizootic. So instead of endemic is enzootic, instead of epidemic, epizootic. Also the panzootic instead of pandemic are all these terms that are used in veterinary medicine. Now, the period also of contagious is another word that we use, which refer to the time during which an infected animal sheds the virus. It can vary between animals. For example, in the case of HIV, it can be shed, the virus can be shed in the absence of any clinical symptoms. So the period of contagiousness is a way, if you want, to measure or to describe virulence or the pathogenesis of a virus. Any questions? Okay, so